Welcome to Biochemistry Unfolded, From Atoms to Algorithms, an online podcast on the topic of biochemistry and biophysics. Well, good morning and welcome to another episode of Biochemistry Unfolded, From Atoms to Algorithms. So today let's talk a little bit about one of the original genetic algorithms that we use to make life, and how did life originate, right? So we've got to go back about 4 billion years ago, and Earth was a barren, rocky wasteland. It had water, but it was missing the instruction manual for life. Then something changed. A rain of fire from the sky, asteroids and comets, pelted the planet for millions of years. Scientists have long suspected that these space rocks didn't just bring destruction, they brought the ingredients for life itself. Scientists have been searching for the building blocks of life in outer space for many years now. They've found amino acids in space. They found bases of DNA, adenine, cytosine, guanine, uracil. But there's always been a missing piece to the puzzle: sugars. Without ribose, you can't have nucleic acids, such as RNA or even DNA. And without glucose, you don't have the fuel for metabolism. For decades, scientists have tried to find them in meteorites on Earth, but there was always a catch. How do we know these sugars didn't just come from terrestrial contamination? So in 2025, just last year, that mystery has been solved. A brand new paper published in Nature Geoscience reveals that the OSIRIS-REx mission didn't just bring back dust. It brought back a sweet surprise. For the first time ever, in the most pristine sample of a carbonaceous asteroid that we've ever touched, scientists have confirmed the presence of the bioessential sugars that make life possible. And so in this episode, I'm going to break down how they found it and why space glucose is a game changer in the RNA World Hypothesis. And then we'll talk about why this discovery might mean that the recipe for life is written into the very chemistry of our solar system. So let's dive in. First, let's talk about the RNA World Hypothesis. What is it? The RNA World Hypothesis is a widely accepted scientific theory that suggests life on Earth began with a single versatile molecule, and that's RNA, or ribonucleic acid. Not DNA, remember RNA is single-stranded. And so in modern biology, life relies on a complex division of labor between DNA, which stores information, as well as proteins, which catalyze those chemical reactions. The RNA World Hypothesis proposes that before either DNA or proteins existed, RNA actually performed both of those roles simultaneously. So RNA was actually the original molecule that stored the genetic code. So now the question is a bit similar to the chicken or the egg hypothesis. Which came first? In order to understand the RNA World Hypothesis, let's first understand the problem. In modern cells, DNA needs proteins or enzymes to replicate itself. And those proteins need DNA to provide the coded instructions for how to make them. Of course, it's unlikely that both DNA and RNA, DNA and proteins originated at the same time. So RNA is sort of like a molecular Swiss Army knife. In the 1960s and 80s, scientists discovered that RNA has two unique properties that allow it to act as both DNA and protein. Like DNA, RNA is made up of a sequence of nucleotides, adenine, cytosine, guanine, and uracil, that can store the genetic code. But unlike DNA, RNA can form into complex 3D shapes. This allows it to act as an enzyme, specifically called a ribozyme, to speed up chemical reactions, including its own replication. So the RNA World Hypothesis suggests a step-by-step evolution of life. First, we have simple organic molecules like the ribose and nucleic nucleobases that were found on the asteroid Bennu, and these were formed in the early solar system or on early Earth. Then these building blocks joined to form short strands of RNA. Eventually, an RNA molecule emerged that could catalyze its own replication, making copies of itself in warm little ponds or near hydrothermal vents. And over time, RNA began to link amino acids together to make proteins, which are better catalysts for enzymatic reactions. Later, the system evolved to use DNA, which is more chemically stable for long-term storage. And so one of the biggest hurdles for the RNA World Hypothesis has been the sugar problem. And so ribose, the sugar that forms the backbone of RNA, is chemically fragile and difficult to synthesize under early Earth conditions in the primordial Earth. So the detection of ribose in the pristine samples from the asteroid Bennu provides a physical evidence that this essential component was present in the early solar system. It suggests that the kits for building an RNA world were being delivered to Earth from space, making the transition from chemistry to biology much more plausible. So in order to learn more about the formation and evolution of the solar system, NASA launched an asteroid sample return mission in 2016 using an unmanned spacecraft, and that was OSIRIS-REx. The spacecraft rendezvoused with asteroid 101955 Bennu on December 3rd, 2018, and spent the next 2 years analyzing the surface to find a suitable site from which to extract a sample. Bennu was chosen because it's kind of a time capsule from the birth of the solar system. Bennu is a carbonaceous asteroid, or it's a C-type, which is the most common variety. And these asteroids are volatile rich and have a low amount of sunlight reflected from their body, and their composition includes a large amount of carbon. Carbon is one of the 6 main elements that commonly form the building blocks of living organisms. So this increases the likelihood of finding potential biomolecules. And so on October 20th, 2020, OSIRIS-REx touched down on Bennu and successfully collected a sample. Then it left Bennu on May 10th, 2021, returning to Earth on September 24th, 2023. And so it then started on its next phase, on an extended mission to another asteroid, 999423 Apophis, where it's expected to arrive in April of 2029. And so the paper that's published in Nature Geosciences that was just published in December 2025. And so this reports the discovery of bioessential sugars, including ribose and glucose. And these are in pristine samples that were returned from asteroid Bennu by OSIRIS-REx. The findings suggest that in all three fundamental building blocks of life, nucleic acid bases, amino acids, and sugars, were present in the early solar system and could have been delivered to prebiotic Earth. The study identified several sugars that are crucial to terrestrial biology. These included ribose, the component of RNA, as well as glucose. That's a primary energy source for metabolism. And of course, other detected sugars included lactose, xylose, arabinose, as well as galactose. And unlike meteorites found on Earth, which faced terrestrial contamination, the Bennu samples were collected and curated under very strictly controlled high purity conditions. So this leads to the absence of significant biological contamination, and this was further supported by finding nearly racemic, that's equal D and L amino acids in the sample. So the distribution of sugars in Bennu is consistent with a Formose type reaction where formaldehyde reacts in aqueous environments to form larger sugars. And so the authors proposed these sugars that were formed within the parent asteroid's brines rather than in interstellar space. So that was fairly significant. The presence of ribose also supports the RNA World Hypothesis, which suggests that RNA was the first informational and catalytic molecule for life. The detection of glucose, which has not been confidently reported in other extraterrestrial materials before, indicates that primitive metabolic substrates were also available in the early solar system. And so how did the scientists do this? So if you look at the paper, the methods section kind of highlights some things. So in the sample analysis, the researchers analyzed a 603.4 milligram aliquot of homogenized Bennu powder. And then these sugars were extracted from this powder using acid and water. Acid extraction was specifically used to prevent sugar degradation or artificial formation during the process. And then they kind of analyzed this extract. And so they analyzed this with gas chromatography and mass spectrometry, that's GCMS. It's a fairly common chemical system. And they also used GC tandem MS, GCMS/MS, and this was used to identify and quantify the sugars. And so you get little peaks on a graph that indicate where the sugars are. And so then these identifications were confirmed across different GC systems and separation columns to confirm accuracy. They used a silicon dioxide procedural blank that was processed alongside the samples to monitor for any laboratory contaminations. They were very careful about looking for that as well. And then they looked at the environment as well. So the team used the pH of hot water extracts from Bennu, finding a fairly weakly alkaline pH of about 8.23 to estimate the chemical conditions on the asteroid's parent body. And so the return samples from asteroid Bennu are exceptionally pristine. The paper actually highlights several limitations and weaknesses, however, that are primarily related to the analytical constraints studying such extraterrestrial material. It's very important to look at both the strengths and the weaknesses just to kind of look at that. You want to try and provide a big picture, right? So some of the weaknesses and limitations, there were a fairly low abundance of sugars detected. The total abundance was about 0.668 nanomoles per gram. And this is only about 1 mole percent of the amino acid concentration that's found in previous Bennu analyses. There was some analytical constraints, and so due to the low sugar concentrations for the study, researchers were actually unable to perform stable isotopic or enantiomeric measurements of the chiralities. So these measurements are often the gold standard for definitively proving extraterrestrial origin by distinguishing the material from terrestrial life. And so they also found the presence of high abundances of other interfering molecules in the sample that further complicated the detection and quantification of the sugars as well. The ribose and glucose were confidently identified, as well as the detection of other hexose or 6-carbon sugars, specifically mannose and altose. But this was not definitive due to overlapping chemical signals in the GCMS, in the gas chromatography mass spec. And there was absence of DNA sugars. Now, DNA and RNA, ribose is just ribose, and then DNA is deoxyribose. It's 2-deoxyribose. And so they did not find any DNA sugars. And so the theory with this is that because it's highly reactive, it was likely consumed by other chemical reactions within the asteroid's parent body long ago. So that was kind of interesting. But they did look at some co-eluting compounds. The gas chromatography mass spec analysis showed additional peaks that were derived from compounds that exited the testing column at the same time as the sugars. And so these kind of obscured some clear readings. So there was a little bit of uncertainty there that they looked at. And there was some chemical stability issues because some detected sugars, such as ribose, are chemically unstable. And so the reason for ribose's relatively high abundance in the Bennu sample, despite this instability, is not yet fully understood, and that requires further investigation. But despite these overall limitations, the study emphasizes that because the samples were collected in space and curated under high purity nitrogen, they are far more reliable than sampling from meteorites found on Earth. And these suffer from uncontrolled exposure to the biosphere. Overall, this idea that the seeds of life arrived from elsewhere in the galaxy or the universe isn't just scientific theory. And so this theme has actually haunted our science fiction and even our theology for decades. So let's take a look at the popular show Battlestar Galactica, either the 1980 version or the reimagined version recently. Now in the show, humanity searches for their ancestral home Kobol. If that sounds familiar to some of you, that's because the show's creator, Glenn A. Larson, was a member of the Church of Jesus Christ of Latter-day Saints. And so he borrowed the name from Kolob, which in Mormon scripture is the star nearest to the throne of God. And of course, Kolob and Kobol are anagrams of each other. Latter-day Saint theology actually proposes a spiritual form of panspermia. And that's the idea that our spirits and even the organized matter of our world originated in the heavens. Whether you're looking through a telescope or a religious text, we've always been obsessed with the idea that we're strangers on this planet. So does this prove the RNA World Hypothesis? Not quite, but it removes a massive roadblock. By showing that ribose can survive the journey through space, it makes it much more likely that RNA-based life could have sparked the Earth's early oceans using imported materials. And so the Bennu samples are the most pristine we've ever had. They tell us that even if we're alone in the universe, the ingredients for neighbors are scattered literally everywhere. And so what do you think? Did life start in a warm little pond, or was it delivered from the stars? And is it just a coincidence that our sci-fi myths look so much like our latest scientific papers? Let me know in the comments down below. And don't forget to like, subscribe, and stay curious. And I'll see you in the next episode. Thank you for watching. Hope you enjoyed. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.